Narendra Deva University of Agriculture and Technology, Kumarganj, Ayodhya. I cordially welcome you all to the second day of this 7 days international training come workshop on future technology in veterinary sciences in Viksit Bharat, agriculture transformation for sustainable livelihood practice. This program is jointly being organized by Vital Biotech, Agriculture Forum for Technical Education of Farming Society, Kota, and College of Veterinary Science and Animal Husbandry, Acharya Narendra Deva, University of Agriculture and Technology, Kumarganj, Ayodhya. Vital Biotech is India's leading publication, research, and training institute, whose goal is to tackle the world's most pressing problems, which related to food and agriculture. In order to achieve this, it is actively involved in the creation and manufacture of microbiological and biotechnological substances and molecules gaining respect both domestically and globally in the field of scientific publication. Vital Biotech aims to publish top-notch, progressive, peer-reviewed research articles, whereas Agriculture Forum for Technical Education of Farming Society, Kota, serves as a spiracle between the network of rural communities in India and other stakeholders, such as the government, development organizations, and corporate entities. It is a spherical organization of agricultural professionals and related sectors in India. It provides a common platform for various agricultural professionals like professors, associate professors, assistant professors, PhD scholars, students, mystics, experts, and researchers working in various scientific institutions across India and abroad. The College of Veterinary Science and Animal Husbandry has been tirelessly working for the betterment of the livestock sector by providing quality education and catering for the needs of farmers in the field of animal production by providing its technical expertise for the genetic improvement of livestock and poultry and better animal health and treatment. The well-learned and up-to-date faculties shape the budding veterinarians of the college by providing the best quality teaching and counseling. And I would also like to announce that our knowledge partners for this program are University of Toledo Medical Center, Ohio, USA, Colliston University of Veterinary and Animal Sciences, Bahawalpur, Pakistan, Lala Lajpat Rai University of Veterinary and Animal Sciences, Hisar, Haryana, Kerala Veterinary and Animal Sciences University, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana, Punjab, Sher Kashmir University of Agricultural Sciences and Technology of Jammu and Kashmir, Kamdhenu University, Gujarat, Bihar Agricultural Sciences University, Patna, Odisha University of Agriculture and Technology, Bhubaneswar, University of Agriculture Sciences, Raichur, Karnataka, Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur, University of Veterinary Sciences, Jaipur, Rajasthan, and Agrosphere e-newsletter. And also, I'd like to congratulate the entire team for organizing this insightful training program. Today, we are highly privileged to have such honorable dignitaries among us. It is a matter of great pride for us that we have Professor Dr. Sushila Man with us to interact with our participants this evening. Madam has joined us. I wholeheartedly welcome you, ma'am. Professor Dr. Sushila Man is currently working as Principal Scientist and Head Department of Animal Biotechnology, College of Veterinary Sciences, Lala Lajpat Rai University of Veterinary and Animal Sciences, Hisar, Haryana. Madam has completed her graduation and post-graduation from Chaudhary Charan Singh Haryana Agricultural University, Hisar, PhD from Royal Veterinary College, University of London, and postdoctorate from the Institute for Animal Health, Pirbright, UK. Madam has vast experience and worked in different capacities which are veterinary surgeon, technical officer, assistant research officer, and principal scientist, and ma'am has been awarded with numerous prestigious awards and honors, which include Dr. B.D. Bhal Gold Medal, Certificate of Honor for Best Academic Record during graduation and post-graduation, Commonwealth Scholarship for pursuing PhD in UK, Postdoctoral Fellowship, Biocare Fellowship, Best Researcher Award, Vigyan Ratna Award from Haryana State Council of Science and Technology, Inspiring Lady Veterinarian Award, Punjab Rao Deshmukh Outstanding Women Scientist Award of ICAR, and many more. Madam has numerous notable publications and is also an academic editor, editorial board member, referee of various international and national journals, and member of several professional committees. Madam is also a fellow of the National Academy of Veterinary Sciences, NAS, Veterinary Research Club, UK, ASM, IVS, AMI, IAVMI, ISVIB, IALV, and many more. Madam, your achievements are endless, and we are not able to enlist them all in such a short time period. So we are very indeed fortunate to have you on board, ma'am. With these words, I would like to hand over this virtual stage to Dr. Sushila Man. Ma'am, over to you, ma'am.
Thank you, Miss Silo, for the lovely introduction and such a elaborative, nice words you said about me. Thank you so much. Good evening to everyone. Before I start, I would like to wholeheartedly like to thank the organizers of this meeting, Vital Biotech, FAs, and the University, Ayodhya University, for giving me the opportunity to work talk at this platform. I am trying to share the screen. Is it possible? Screen share, but it's not allowing me. Is there? How do we share, Miss Silo? Can you help me? Because it gives me a question.
Sir, can the slide be sent to you and shared?
I had shared earlier, but it is not allowing today. I don't know why.
Oh,
Okay. Ma'am, first open your PPT on your desktop and
It's
then go
yeah,
to the screen share. Then you will find the PPT in your desktop and you can share from that area.
yeah, that's what I'm trying to do. Or else I send it to someone now. Shall I send it to somebody? If someone can tell me the email address, Mehta Saab, can you tell me the address?
now, sir, can you please share your email address so that ma'am can send
Yes,
the mail to you?
yes, ma'am.
Yeah, please speak out.
Yeah, just a minute, ma'am.
Yeah. But it is exceeding the size, so it will not allow me. Sorry about that. I don't know why it's not allowing. Maybe some version problem or
Ma'am, is it open in your background,
it
the
is
PPT?
in my background and it is open.
Oh,
Yeah.
ma'am, maybe you can send it to me, to my mail. I will write my mail in the chat box.
In the chat box.
Yes, ma'am. Ma'am, I've dropped my mail in the chat box, so if you can,
Yeah.
I will download from my mail and get back to you.
Meeting chat. Even I cannot see the. Yeah, it's here now. Dr. Krishan Nama.
Yes, ma'am. It's my mail ID. If possible, then please mail your PPT to on my mail ID. Then I will share it from my side.
Let's
Yes, ma'am.
I never expected it will happen like this because normally I do the Zoom meetings.
No problem. Huh? No problem.
No problem, ma'am.
Yeah. Only problem is it's taking time, so we'll run out of time.
While we're waiting for ma'am to share her slide, I would like to request Dr. Jitendra Mehta, who is the course director for this training and workshop. He is currently the president of AFTFS, this organizing team. So a research scientist and a dietitian. So I would like to request sir to share a few words about whatever you would like to share about this workshop training. Sir, please.
Hello, everyone. This type problem will come, and as you know about vital biotech publication, we are working last 10 years, and we have 4 journals and 4 magazines, and vital biotech publication also published books in various fields, maximum books from agriculture and veterinary sciences, and we are successfully conduct lot of events in last year, and very soon we will update this PPTs also. Just a minute.
So dear participants, good evening to all. I am Dr. Dharmesh Tiwari, Assistant Professor in Acharya Narendra University.
Sorry for interruption.
No problem, sir, no problem.
Vital Biotech has started 2009 with 9 students, and this journey is also very excited. Like only 9 students is there. They want for training. They are searching, and at that time, I told them like you can do mushroom production. So they are already searching. Then only just a minute.
Tiwari sir, also sir, if you'd like to share some while sir is not available.
Dr. Nama, could you got my talk on the Google Drive link because the file is heavy? Could you open it, Dr. Nama?
No problem, ma'am. He is doing something. He will share, ma'am.
Okay, it's fine. There is some version problem, I guess, on my laptop, the Zoom version problem, because it gives question mark when I do the share screen.
Okay, ma'am. Actually, this is technical glitch, ma'am. Nobody can do anything. And you can say it's a positive sign. Our maximum participants are joined, ma'am. Initially, there is only 30 or 40 participants. Now we reach to the 100 participants.
Okay.
So everything is going for good.
That's true.
जो भी हो रहा है अच्छे के लिए हो रहा है. ज्यादा पार्टिसिपेंट मैम आपको सुन पाएंगे, नहीं तो आपका बहुत इंपॉर्टेंट जो भी लेक्चर था ये शायद पास हो जाता, बहुत लोगों से मिस हो जाता. तो मैम एक रिनाउंड एनिमल बायो टेक्नोलॉजिस्ट हैं. मैं अपने पार्टिसिपेंट्स को बताना चाहूँगा. और टुडेज लेक्चर इज वेरी मतलब फ्रूटफुल फॉर वेटनरी स्टूडेंट्स एंड साइंस. मैम का जो एक्सपोजर है वो इंडिया के साथ साथ एब्रॉड में भी है, तो हो सकता है ये आपके लिए फ्यूचर में कहीं ना कहीं काम आए. बिकॉज देयर इज नो एनी रिसर्च इन वेटनरी साइंस विदाउट एनी एनिमल बायोटेक्नोलॉजी. देयर इज ऑलवेज यूज सम मेथड्स एंड सम parameters which is attached to the animal biotechnology. It's a very nice field, very creative field, and our speaker is also very renowned, and we can we can capture her experience, so we can change our life. So don't be परेशान मत हो सारे participants. Be add और अपने और अभी साथियों से कहें. We are 100 and our participants are ma'am nearly 250. Okay. So only half has joined here. So more participants will be there. It's a more fruitful for others one. So ma'am, today is ma'am and other participants. Today is total 3 PowerPoints or presentation or speakers are here. One is ma'am, and another one is a renowned pharmacologist, head of Jammu Veterinary College, and the third one is from my college, my colleague Dr. V.K. Paul, and the second one is Dr. Pankaj from Jammu Veterinary College. So it's a very, you can say, very capturing day for all participants. We can give the three field: animal biotechnology, pharmacology, and parasitology. Because our third speaker, Dr. V.K. Pal, is an assistant professor in Department of Parasitology. So we can, in one day, we can capture, or we can study, we can take the experience of three different persons and different subject of veterinary science or veterinary field, which can help to enhance your knowledge and some others. So ma'am is one pharmacologist from Jammu, another one, and the third one is from parasitologist. I think there is, ma'am, presentation PPT will be, I think, is very large. That's why there is a problem is going on.
Okay. Let me start the on, but you cannot see the screen. This is the, no, but I used it before. There is, I cannot say anything. Why it is not allowing me?
No, ma'am, it's going started, ma'am. Don't worry.
Starting.
Yes, ma'am.
Dr. Nama, so nice of you. Only thing is, screen is not in my hand. So there's no problem. It's okay. Shall I start now?
Yes,
ma'am,
ma'am.
please.
Please carry on.
If you can do it on the full screen, please. Screen slideshow. Yeah. Okay. Good evening, everyone, once again, and thank you, Dr. Ms. Silo, for the introduction, and Dr. Tiwari and Dr. Mehta for filling up the gap. I'm really sorry, and I apologize for this time lapse because our time is limited. So, first of all, before I start, I would like to thank all the organizers for organizing this useful meeting and giving me the opportunity to talk here. So I will talk today on novel vaccine technologies in veterinary sciences. We all, particularly veterinary graduates, and we all know who are working in the veterinary field, that the success of traditional vaccines are inactivated and live attenuated vaccines. They have enhanced the livestock health, productivity, and promoted food security, and reduced the morbidity and mortality due to several human and animal and zoonotic diseases. But these traditional vaccines, they are not without fault. They have one or two inherent disadvantages. Like inactivated vaccine, they generate poor immune response, so they need booster. On the other hand, live attenuated vaccines, they have the problem of reversion back to virulence. So the traditional vaccine have some of the inherent disadvantages also, apart from their advantages. And because we know that diseases are emerging day by day, and COVID-19 is one of the biggest example. And we need to quickly deploy the vaccines to control these diseases. But unfortunately, the first generation vaccines, they are not conducive to such urgencies. So there is a need for development of new vaccine strategies. And in the last 3 decades, there is a lot of advancement in the veterinary field, in the particularly in the field of vaccine development. And my this talk I have organized to give you an overview of the available novel vaccine technologies which are currently being utilized in the companion animal, food animal, and wildlife disease control. Next, please. So, little bit of introduction. The term vaccine has derived from a Latin word called vacca, which means cow. So the Edward Jenner, who is the father of immunology, he described the inoculation of human being with the cowpox vaccine. He used related virus, cowpox virus, to immunize the human being. And in this way he developed the first vaccine because he observed that milkmaids who got the cowpox infection, they were immune to the smallpox. So that is how he developed the first vaccine. He used to vaccinate against smallpox using the cowpox vaccine. So likewise, he and that's why he is known as the father of immunology, The another important figure in this field is Louis Pasteur, who is the father of microbiology, but he is the progenitor of modern immunology also, because he popularized the germ theory of disease. And he postulated that the infectious diseases, they are caused by germs, and they could be prevented by the prophylactic vaccination. Before his this concept, the spontaneous generation theory was there for the germs. That means people used to think that germs come out of nothing, but he discarded this spontaneous generation theory and give this theory that diseases are caused by uh, microorganisms. He gave the concept of attenuation or weakening of microorganism, and this way he developed some of the world's first vaccines, like against fowl, cholera, anthrax, and rabies. These are the important diseases in the veterinary field. He developed the method of pasteurization. Also, the technology is named after his name, where you heat the liquid to a high temperature, like 121 degrees for certain time, like 15 minutes, to get rid of harmful organisms. So, like this is with this history, we can define the vaccine as a biological product that can be used to safely induce an immune response to confer protection against any infection or the disease on subsequent exposure to a pathogen. To achieve this, the vaccine must contain antigens that are either derived from the pathogen itself, or they can be produced synthetically to represent the component of the pathogen. So, to summarize, from Edward Jenner to Louis Pasteur, from 18th to 19th century, till the eradication of rinderpest, an important disease of bovine, and the smallpox in humans by 20th century, the vaccines have played a vital role in the survival, in the health and general well-being of human and animals. Next, please. So, a goal of vaccination is to generate humoral or cell-mediated immunity, thereby inducing the production of immunological memory and that confer protection against the subsequent natural infections. Although we know that the goal of vaccination is to provide neutralizing antibody against that particular pathogen, but apart from neutralizing antibody, the other arm of immunology, that is called as cell-mediated immune response or the cell-mediated immunity, this is also crucial for protection against certain pathogens. Next, please, like varicella, HIV, tuberculosis, malaria. So, just few of the introductory slide. There are two types of specific immunity. One is humoral immunity, that is also called as antibody-mediated immunity, and this is based upon the production of antibody and the antibody activity. The other is cell-mediated immunity or cellular immunity, and this is based upon the specific kind of T lymphocyte. What happens here, if you see in the diagram, the complex antigen, this is processed by phagocytes, and it is presented to B and T cell. And once the antigen is processed by certain phagocyte, then the humoral arm and the cellular arm of the immune response get activated. So once the humoral arm, the B cell, and the cellular arm, the T cell, they are activated, they proliferate into effector, memory, and the effector cells. If you see the B cells, memory B cell and plasma cell, specific plasma cell, they will secrete specific antibody in response to a infection or an antigen. While the memory cell, they keep the memory, and once the host will be exposed to that antigen again, this memory cell will get activated and they will produce the specific antibody again. Similarly, if you look at the cellular arm, the T cells, the effector cell will produce the different kind of cytokines like interferon and others, while the memory T cell gets activated like the memory B cell once the host will get exposure to that antigen again. Next, please. So another is a type of specific immunity. This is one is innate immunity, the general or the primary defense. Then another is adaptive or acquired immunity. So here it is two types, natural and artificial immunity. Natural is also active and the passive. Natural immunity is acquired by an individual in the normal life experience, and it is not induced by any medical means. Active immunity, once the person is exposed to a infection, then his or her body will generate immune response to that particular microbe, and that is how he will acquire the active immunity. And the passive immunity, like maternal antibody, this is the consequence of a person receiving the preform antibody made from another person. So these are the natural ways. Then artificial immunity, this is produced through medical procedure, or we call it as immunization. So we give a person will acquire active immunity if we get a vaccine against that particular microbe. So this is by vaccination. Then passive immunity is a consequence of a person receiving preformed antibodies, like immune globulin therapy. We have seen in the COVID also people received the immune globulin therapy. In the rabies also we get the post-bite immunoglobulins. This is how the natural and acquired immunity is there. Artificial immunity is there. Next, please. So these are the types of vaccines, the first generation, second and third generation vaccine. First generation is vaccine is the traditional, our classical vaccine. These are the whole virus vaccine, either two types, inactivated and the live attenuated. Inactivated means we kill the virus by certain procedures with certain chemicals, and live attenuated is we weaken the pathogen using certain procedure. Either we go multiple passage in the cell culture or other means. Whole virus vaccine, you are inoculating complete virus in different formats. Then second generation vaccine include protein-based vaccine, subunit vaccine, and viral vectored vaccine. Viral vectored vaccine are also two types. I'll come to them in one slide each and explain to the house. The non-replicating viral vectored vaccine and the replicating viral vectored vaccine. Then the third generation are there, nucleic acid vaccine that has DNA and mRNA vaccine, and then nanoparticle based vaccine. Next, please. This graph is another format of different vaccine platform. So these are the different vaccine platform we have currently in the veterinary medicine and even in the human also. The veterinary development is very important because that leads to the development of human vaccine also, like the same inactivated, live attenuated, virus-like particle vaccine, viral vectored vaccine, subunit vaccine, synthetic peptide vaccine, DNA vaccine, and mRNA vaccine. Next, please. So the first, and let's talk in very brief about what are the first generation vaccines. So live attenuated vaccine, they contain whole bacteria or virus which have been attenuated by certain procedure, and they produce a protective immune response, but they do not cause disease in the healthy individual. So some of the vaccine example I have written here, which are live attenuated, and they are globally used for important diseases like rotavirus vaccine, MMR vaccine, measles, mumps, rubella vaccine, nasal flu vaccine, shingles, chickenpox, and BCG vaccine. Next, please. Then the inactivated vaccines are there. Next, please. If you can give two tabs, please. Another. Next, please. Yeah. Inactivated vaccines. Previous slide, please. And the inactivated vaccine, they contain whole virus or bacteria which are killed or they have been altered so that they cannot replicate. They cannot cause the disease even in the immunocompromised individual. So they are very safe to use, but they do not they cannot create very strong or long lasting immune response as compared to their live attenuated counterparts. So they need boosters. So they give 6 months or 1 year immunity, and then you need a booster. Some of the important vaccines which are inactivated, they are polio vaccine, some of the flu vaccine, and hepatitis A vaccine. Again, like other platforms, they have advantage, disadvantages. They have, both of them have the ability to stimulate the innate immunity. Innate immunity means the general immune response, not specific. So they induce long-term protection, and they are very easy and cost-effective to produce. These advantages I already told, like inactivated, they need booster, and for live attenuated, they have the problem of reversion back to virulence. Sometime For example, for the segmented viruses like rotavirus, they have the 11 segments of their genome, and the blue tongue viruses, they have 10 segments in their genome. So if a host is infected with a wild type virus and the host gets live attenuated vaccine also, so their genome can reassort, so they can exchange the genome segment because they are like segmented genome, so they have the potential of reversion back to virulence, which can be dangerous. Next please. Now the second generation vaccines, they include, next slide please. The second generation vaccines, they include the subunit elements of a pathogen, conjugated or recombinant antigen, synthetic protein, non-protein antigen, or expressed bacterial proteins, which include numerous molecule and epitope of different species and strains of pathogens. So different parts of pathogen, either natural or synthetic, they can be used as a vaccine. These are the second generation vaccine. So I'll come to them one by one in few slides, coming slides. But they have certain advantages because they are cross-reactive, cost-effective. They are very safe to use because they do not contain any live or attenuated organism. So they cannot revert back to virulence. They cannot cause the disease even in the immunocompromised individual. And they are very effective because they produce stronger immune response. And most importantly, they can produce both humoral and cell-mediated immunity. This is very, very important because in certain pathogen, humoral immunity alone cannot serve the purpose, and you need a cell-mediated immunity also. So they generate both type of immunity. Disadvantages because they immunogenicity because they do not contain pathogen-associated molecular patterns, the PAMPs, which are required by immune response, immune system to recognize the pathogen by uh, pathogen recognition receptors. So because they lack the PAMPs, so they need repeated vaccination and they need adjuvants because they lack PAMPs, so they need adjuvants. Next, please. The next slide, please. Next slide, please, Dr. Nama. So third generation vaccines, they include the nucleic acid based vaccines. They are the DNA RNA vaccine, vectored vaccine, and live attenuated or inactivated chimeric vaccines. Because we have the advancement in molecular techniques now, so we can manipulate these nucleic acid of the pathogen to our advantage. Thus, we can develop the new vaccine, which are alternative to classical technologies. So this DNA RNA vaccine, this is a new approach. Next, please. This is a new approach where you clone the immunogenic part of a pathogen nucleic acid into a plasmid. You can clone the antigen of interest into plasmid and they can be delivered directly through injection. Yeah, there are several advantages of these third generation vaccines in immunocompromised individuals, unlike modified live virus, the attenuated vaccines, which can be problematic. So they have the potential of, here you can combine multiple plasmids and you can get a broad spectrum combined vaccine. So they are very easy to engineer. Then they can induce both humoral and cell-mediated immune response. A very important part is they can function as PAMPs, I already told, thereby reducing the need for adjuvants because using with this vaccine, we don't need to use adjuvants in majority of the cases. And also they can initiate the cell-mediated immune response also by activating the MSC class 1 and class 2 complexes. Thereby they can activate the CD8 cell responses and CD4 responses. CD8 by activating the MHC class 1 and by CD4 by activating the MSC class 2 molecules. Next, please. So another area is the recombinant vaccines. They are produced by inserting the RNA, DNA genes into the some expression vector like yeast cell or viruses, and it results in the production of specific component of the original pathogen, and the antigen is expressed in the cells. They can be purified and used as a vaccine. So these are three categories here. The subunit recombinant vaccines. These are the component of pathogenic organism. They include the subunit protein vaccine, peptide vaccine, multiple peptide vaccine, and the DNA vaccine. Then the attenuated recombinant vaccine. Here they are genetically modified organism, either bacteria or virus. They are made non-pathogenic and can be used as a vaccine. The third category of recombinant vaccine is vector recombinant vaccine, where genetically modified viral vectors you take like pox viruses and like big viruses, you can delete their genome and insert the gene of your interest, and these viruses can be used as a vectors to deliver the antigen of interest against certain pathogens. Next, please. So recombinant subunit vaccines, as I said earlier, these are made from a very short specific protein of a pathogen that are non-infectious. So they cannot replicate in the host, so they are very safe to use. So some important subunit vaccine are polysaccharide vaccine, conjugate vaccine, and protein-based vaccine. Here, as the diagram shows, the pathogen genome is sequenced, the gene of interest is identified, that is cloned, and this recombinant construct can be used to generate the DNA vaccine, subunit vaccine, or vectored vaccine. So, like here, the recombinant protein vaccine, they can be made in either expressed in bacteria or the yeast, and they can be expressed in this system. Next, please. One of the biggest example of this recombinant vaccine is hepatitis B vaccine, which is very successful because surface protein, this is hepatitis B surface antigen, HBS antigen. This is very immunogenic and very useful as a vaccine candidate. This is expressed in a powerful and cheap expression system. That is, expression system is a methylotropic yeast named as Pichia pastoris. This is very good expression system because this system facilitates the genetic modification. It allows the secretion of expressed protein. It permits the post-translational modification, and it produces the expressed antigen is produced in a very high quantity. And this hepatitis surface B antigen is expressed in Pichia pastoris, and this is very useful. It is very highly immunogenic, very effective and powerful response it generates against the hepatitis B virus. And there is no genome, so there is no risk of multiplication of the virus. So other similar vaccines are anti-herpes simplex virus vaccine, anti-rotavirus vaccine, hepatitis B virus vaccine, using the same system expression in the yeast system. So these are available in the market, these vaccine against these important viruses. Next, please. So as I said earlier, there are toxoid vaccines. They are one of the example is the DPT vaccine, diphtheria pertussis tetanus vaccine. Some you know that some bacteria they release very poisonous protein which are called as toxin. So here we will want to target this toxin for production of vaccine rather than bacteria, because bacteria itself is not dangerous, but the toxin is dangerous. So this immune system here recognize the toxin in the same way as it recognize the other antigens on the bacteria. So these vaccines are made from with the inactivated version of this toxin, and we call them as toxoid because they are inactivated, non-dangerous. We call them as toxoid vaccines. They look like toxin, but they are not pathogenic, not poisonous. So they trigger a strong immune response. DPT vaccine is one of the example of toxoid vaccine. Next, please. Then the conjugate vaccine. Conjugate means the joined. So some bacteria, they get protection from a, you need, for some bacteria, you need to train your immune system to respond to several of the polysaccharides which are available on the cell membrane, cell wall of bacteria. These polysaccharides are the complex sugars on the surface of bacteria. So not a single protein, but you need, your immune system should recognize several of these complex sugars called polysaccharide. So researchers have shown that these polysaccharide, they work much better if they are attached to something else to create a strong immune response. What is this something else is? This is, as I told in the previous slide, the toxoid vaccines. So these polysaccharides, they are combined with the toxoids. So our immune system will recognize this toxoid and generate a strong immune response. This is very important vaccine. If you go this is important one is children pneumococcal vaccine. Immunocompromised individual also take this vaccine. So what is this? It contains polysaccharides from 13 different type of bacteria which cause pneumonia. And these 13 type of polysaccharide, they are joined to a toxoid. And this is how this vaccine is available in market for use in children against the pneumococcal disease to prevent pneumonia. And similarly, other vaccine is this MAN-ACWY that contain 4 type of polysaccharide from 4 different bacteria joined with the diphtheria or tetanus toxoid, and it is used to prevent meningococcal disease. These bacteria, they cause meningitis. So to prevent this meningococcal disease, this type of conjugate vaccine are being used, and they are available in the market in for humans also. And for, next please. Then the next sort of platform is, next slide please. Virus-like particle vaccine, very important vaccine. I'm sorry for the busy slide, but I have simplified this. These virus-like particles are the molecules, they resemble the virus, but they are non-infectious. They have the virus coat only and with no genetic material. So they are naturally, you can take them from the natural system, or you can synthesize them artificially. You synthesize all the viral outer capsid structural protein without, and then do the assembly in vitro in the lab and use them as a vaccine candidate, because they will mimic the complete virus. In the diagram here, this is the paper from Poleroy et al. Poleroy and her lab in 2009. This is regarding the blue tongue virus. Blue tongue virus is the virus I have worked, one of the arbovirus, and it causes disease in small ruminant and even in the cattle. I have worked on this virus for more than 25 years now. So you see here, this Poleroy is a very renowned personality in the field of blue tongue research. She had made this virus-like particle vaccine where she has synthesized all the protein. In the dark, in the panel below, you see VP3 alone. VP3 is the inner capsid protein. Then in the panel in the middle is the VP7, the another inner coat protein, and the assembled is VP3 and VP7 is the core-like particle. So they will give protection against whole of the 30 serotype of blue tongue virus. They are generic viruses, the core-like particle, because VP3 and VP7, they are the conserved protein. So you will get protection against all the 30 strain serotypes of blue tongue virus. While if you add the outer coat protein here, the third panel, VP3, VP7, VP2, and VP5. VP2 and VP5 are the outer coat protein. She has done co-expression of all these protein in the baculovirus, the eukaryotic expression system, and allowed the virus to assemble. And when the protein are assembled, you can see in the cartoon above, these look like the complete virus particle without any nucleic acid, and they can protect against the specific serotype depending upon which serotype VP2 and VP7 protein, VP5 protein you have taken. So very few virus-like protein-based vaccines are available in the market: hepatitis B vaccine, human papillomavirus vaccine, and the blue tongue virus vaccine. Only disadvantage of VLPs are they are very costly to produce and their shelf life is a bit low. So you have to keep them, the cold chain need to be maintained for making the vaccine stable, keeping the vaccine stable. Next, please. Next slide, please. Then the outer membrane vesicle vaccines, I have taken this from a recent paper of 2022 from Frontier in Microbiology. These outer membrane vesicle vaccines, they are naturally produced by bacterial outer cell membrane. They contain many of the antigen that are found on the cell membrane, but it is non-infectious. In the lab, these OMVs can be harvested from bacteria and they can be used as a vaccine. They can be modified in the lab so that the toxic antigens are removed and only antigens which are suitable for stimulating the immune response and non-toxic, they can be kept. They are natural adjuvants. You don't need adjuvant with this kind of vaccine. This is a very new vaccine technology and there are very few vaccines they are licensed. One of the example is meningococcal B vaccine. I previously mentioned this is like the conjugated vaccine and this is the OMV vaccine against meningococcal B, um, meningococcal bacteria. Next, please. Then the like every other any other platform, there are pros and cons of every system. So this subunit vaccine, they have the advantages they can incorporate the protein in the native form. So the protein will be there in the correct folding. So the epitope will be in their natural format and they are in the conformation. So conformational epitope will be there and they can invoke immunity to more than one strain or serotype of bacteria, virus, or pathogen because you can generate the chimeric system. You can use different antigens in the same system. Only problem is they only give a moderate level of immunogenicity because they require adjuvant to generate strong immune responses. Some of the vaccines of veterinary use, they are licensed, which are subunit vaccine. They are for canine leishmania, for leishmaniasis. And we have two type of recombinant protein vaccines are available for leishmania and chimeric protein vaccine is available. Similarly, for swines, for Actinobacillus pleuropneumoniae, the vaccine subunit recombinant protein vaccine is there, and for porcine circovirus 2, and for classical swine fever. These are the two very important viruses circulating in the swine population. Even we have found in swine population of Haryana recently through one of the project. Next, please. Next slide, please. Now the even newer technology, that is the DNA vaccine. Here what happens is gene of interest is taken. This is cloned into expression plasmid. Then we this expression plasmid is used to transform the bacteria, and then we purified the plasmid DNA, and straightway we immunize this plasmid DNA into the host. So this DNA vaccine, they produce antigen in the host itself. Once the plasmid is transfected into the host cell, it is transcribed into mRNA and then it is translated into relevant protein. And now the host cell machinery will produce that antigenic protein itself. So host will produce that required protein itself. The host immune system recognize the antigen as foreign, and it will lead to the development of both humoral and cellular immune response. And for animal use, DNA vaccine are available for West Nile virus in horses. Next, please. Now there are advantages of DNA vaccine because they are very safe compared to live vaccine, And also there is no vector immunity. I will come to that later on. When we use the viral vectored vaccine, you will get immunity against that vector also. So after the booster, you will get lot of antibodies against the vector along with your specific antigen. But in the DNA vaccine, this is not the case. You will get the immunity against that particular antigen. So they promote the induction of CTL response. Also, they're very stable, so you don't need cold chain. One of the example vaccine is from the salmo, salmonid fish against the infectious hepatopoietic necrotic virus and the alpha herpes virus subtype 3. This is from the food animal from the fish. These two pathogens, we use the DNA vaccine. They are available in the market. And here it is written that you from the which manufacturer, what is the brand name of the vaccine and what plasmid they have used. Next, please. So the RNA vaccine. The RNA vaccine, we have in the RNA vaccine we use mRNA that is inside a lipid or fat membrane. We immunize with the RNA, the mRNA that is inside the envelope of a lipid membrane, because this lipid membrane, it will allow the mRNA to enter into the body, and also it will help to get inside the cell by fusing with the cell membrane. So once mRNA is inside the cell, the host machinery of the cell will translate into the antigen protein, and this mRNA will last for only few days. And once the sufficient antigen is made to stimulate the immune response, this mRNA is naturally broken down and removed from the body system. This mRNA vaccine, they are not capable of combining with the human DNA. So there is no danger of incorporation of this foreign mRNA into human DNA. So they are safe to use. Very, very important example is the Pfizer BioNTech and the Moderna COVID-19 vaccine, which were used in Europe. They were the RNA vaccine. This diagram here shows how the mRNA while endosome enters the cell and host cell machinery translate into the different protein, and this is how different TLRLs and other immune system gets activated, and finally the mRNA is removed from the body system once enough antigen is there. Next, please. Now the another important platform is viral vectored vaccine. So as I mentioned earlier, we use viral vectored vaccine by using harmless viruses to deliver the genetic code of the target antigen to the cells of the body. So they can produce protein antigen and stimulate the immune system. The large viruses like pox viruses, canary pox, fowl pox. The pox is the biggest virus. So we take the pox virus genome, remove its pathogenic component, and because of the large size, it can very easily incorporate the foreign genetic material into it. So so we can use them as a vector. So we incorporate our gene of interest against the antigen we want to produce the immune response into the system, into these virus vectors, and they are then grown in the cell lines, and they can quickly and easily can grow to a large scale. They are very cheaper to produce compared to many of the nucleic acid vaccine and many of the subunit vaccine. There are two categories here, replicating and non-replicating. We can use the replicating virus where the vectors retain the ability to make new virus particle. Like if you are using canary pox virus as a vector, so we don't remove its replication genes. So we will incorporate our own gene of interest by keeping the replicating gene of the canary pox virus. So while growing, it will keep on, while making your protein of interest against the known virus, it will keep on generating new pox virus particle also. So the advantage of here is the live attenuated whole pathogen vaccine. They have advantage that they are constant source of vaccine production over extended period of time, because the new virus particle are being generated. So you will get a stronger immune response, and single dose of vaccine is enough to give you protection. Example is Ebola virus vaccine. Ebola virus gene has been incorporated into vesicular stomatitis virus. So then the non-replicating, here the replicating gene of the carrier virus have been deleted. So they lose the ability to make new virus particle. So here the vaccine can be only produced for as long as the initial vaccine remain in the infected cell. So immune response here in the non-replicating case is weaker than the replicating counterparts, and the booster dose is required. So example here is the again Ebola virus vaccine, another kind of vaccine, which was licensed by European Medical Agency in July 2022 after Ebola outbreak in certain areas of the world. And Oxford AstraZeneca COVID-19 vaccine, that is also uh, uh, generated by using non-replicating viral vectors, and this was medical health regulatory agencies in the UK. They have licensed this vaccine for use during COVID-19. Next, please. Now, this is how, just a diagram just to show how the viral vectored vaccine, this is the virus vector on your top left with the red spikes, which are your gene of interest. So it goes inside, and the transcription and translation of antigenic protein take place. As I mentioned earlier, with the DNA vaccine, here there will be immune response. See the step 5 antigen presentation by macrophage antigen presenting cell. You will get antibodies against the carrier vector also. So if you are using the booster with the same vaccine, you will get equal amount of your antibodies against your carrier virus and your antigen of interest. So using the vectored vaccine as a vaccine, so you need a booster with something else. Otherwise, the vector antibodies will, in subsequent boosters, you will get more vector carrier antibodies against carrier virus than your antigen of interest. This is the major drawback of this vectored vaccine. Next, please. So this is the diagram. One more tab, please. This is the diagram representing the formation of vector vaccine. Here, this is the adenovirus vector, adenovirus or alpha virus. These are also big virus. They can be act as a carrier virus. And influenza HA protein is incorporated, expressed inside, incorporated into these carrier vectors, and this is the vaccine against the HA protein of influenza virus. Next, please. Lot of vectored vaccines are available in the market, like for canine, using the canary pox virus vectored vaccine. You have the vaccine against feline leukemia virus, canine distemper virus, and rabies. For equine, using the same canary pox virus as a vector, equine influenza West Nile virus. For swine, these are the chimeric viral vectors, PC, porcine circovirus 2, and classical swine fever. For RNA vaccine is in the swine against porcine endemic diarrhea virus and for swine influenza. For bovine, adenovirus vectored vaccine are available for FMDV. For avian, fowl-pox vectored vaccine are there for avian influenza, NDV, ILT, infectious laryngiotracheitis. And for wildlife, chimeric vectored viral vaccines are there for rabies. Next, please. Next slide, please. Now the DIVA vaccines. Yeah, the previous slide, please. DIVA vaccines. So DIVA vaccines, they and their companion test, they were first developed by colleagues, the Oshorten's colleagues at CBI Lelystad, the Netherlands. Netherlands. These are called earlier before 1999. They are called as marker vaccines. But after 99, they are called as DIVA vaccine. DIVA means differentiation of infected from vaccinated animals. So you here we you here what you do is you make deletion. You delete a particular gene, and you make this deleted gene vaccine. Example is IBR, infectious bovine rhinotracheitis, which is a genetically transmitted disease in bovines, very important disease. And semen has to be tested for this IBR before using it in artificial insemination. So this DIVA vaccine is available. The glycoprotein E gene of the IBR virus has been deleted, and subsequently tests have been developed to differentiate whether the animal is vaccinated or it has got natural infection, because the GE gene blocking ELISA is there, and GE gene based PCR amplification technique is there, so that we can differentiate whether it has received the animal has received the glycoprotein E deleted vaccine or it has got natural infection. Similar to IBR, we have the pseudo rabies, classical swine fever, and FMDV vaccine, which are DIVA vaccines available in the market for bovines. For canine, for leishmania, again, we have the DIVA vaccine. For feline leukemia virus. For swine, for swine herpes virus 1, swine herpes virus 1, and for classical swine fever, we have the DIVA vaccine available. For bovine FMDV and this bovine herpes virus 1 IBR, there is a DIVA vaccine platform available. This is a very important kind of vaccine. You need to know sometime in the field whether you have vaccinated the animal or whether it has got natural infection. Next, please. Next is your nanoparticle or microparticle based vaccine. So because I have worked on orbiviruses, so I will take the example of orbiviruses only. So here, very recent paper from Javier Ortego group from Spain in 2022, they have developed a technique to for three important orbiviruses: blue tongue for small ruminants, African horse sickness for equines, EHDV epizootic hemorrhagic disease virus of deer. They have generated nano and micro and particulated vaccine candidate against these three relevant orbiviruses, and they have developed the innovative technologies for antigen delivery, where they have used the non-structural protein of a avian reovirus and the blue tongue virus NS1, which this protein make tubules inside the infected cell. So these two non-structural protein they have taken for delivery of the vaccine to the right place. And this cartoon, this is I have taken very quickly from their paper. So this cartoon just to show how the vaccine goes inside the cell and how by the lymphatics it is cleared off from the body. And in the B panel, it is show how it activate the different arms of the immune system. So without going into detail of this because of the time limitation, we'll move further. Next slide, please. So by giving the overview to you about all these vaccine platforms, I will tell you in very brief what we have done. We had actually one Indo-UK project from that was funded by DBT, Government of India, and BBSRC, UK, and that was on the farmed animal diseases on arboviruses of livestock importance. This in from 2014 to 18 we had this project. So we had three labs, partnering labs from the UK and five labs from India. Professor Peter Martins from Purbright Institute were the oral coordinator, and I was the Indian lead. Next, please. So I tell you one vaccine platform that we have developed in this project. Next, please, very quickly. So blue tongue is the disease. This is very important for the small ruminant. You see here, you have the oral lesions, the blue tongue, pneumonia, and you have acute diarrhea. And the disease in the southern India, breed of sheep and goat, are very different from that of North India. Next, please. In the North India, you have, these are the lesions. You see the typical blue tongue lesions around the nostrils and the wool break. And this is OI listed disease. And after FMD, This comes under the list of economic important diseases. Next, please. So, what was there here is there were three options of the vaccine when we had this project. We developed the diagnostic, we identified different strains that are circulating in India, and finally came to the vaccines. So very quickly, I will cover here due to time limitation. There are three options: live attenuated. Again, they were made for the South African sheep, and they were causing the pyrexia and viremia, and they were transmitted when they were used in the European and Indian breed of sheep. Then the inactivated vaccine, they are being used in Europe and other parts of the world and even in India, either in the monovalent format and in the bivalent or multivalent format. And then the next generation vaccine, though I talked about virus like particles, but they are not available in the market as yet. Next, please. So you will have to press the button bit quickly here. So in the India, we had 11 centers, including our university. This is BTV network project. So with the network project, we had developed a pentavalent vaccine. Some, not me, other colleagues were there in the department. They developed this vaccine along with other centers, and they were commercialized. They were being used in the sheep and in the southern states of India. They were inactivated vaccine, and they have shown the protection. But every time there is the circulation of another serotype in the region, you have vaccinated against 1 to 10, 16, and 23. Next time you will see BTV 5 or BTV 3 or something else. So we needed a better vaccine. The background is like that. Next, please. So what was there is next, please. Uh, do a two tabs, please. So there was a need to generate a strong protective immune response to protect the animals and prevent transmission. And we needed a vaccine that is DIVA compatible. And desirable characteristics were it should be cross protective against multiple serotype, it should be stable, inexpensive, easy to purify, without requiring the sterile condition, and single injection should be sufficient. These were the requirements of a blue tongue vaccine that was needed to develop. Next, please. So, next slide, please. If you do 3, 4 tabs, please. So, the bacterial expressed protein. So, one more, please. One more. Yeah. Hold on here. So bacterial expressed protein, we all know that they require sterile condition. There is a loss of conformational epitope and cross reaction with the contaminated bacterial protein. So these are the limitation of bacterial expressed protein. Then then the vectored vaccine I talked earlier in detail. So we used earlier people have used modified vaccinia ankara, adenovirus or canary pox for expressing VP2 and other viral protein. But again, you will need sterile condition. You will get antibodies against the carrier virus because genetically modified organism. So you will have licensing difficulties. So these are the limitations. Then recombinant baculovirus, virus-like particle, as I already said, they also require sterile condition, very expensive to make and unstable. They have poor shelf life. DNA vaccine, again, you need uh, sterile condition, they have uncertain efficacies, licensing difficulty. So we went on to plant expressed VP2 proteins. VP2 is the immunogenic outer capsid protein of the virus. So by using this platform, we could generate non-sterile production, very easy to produce in large quantity in the native conformation, very inexpensive and stable to use. You can easily purify them. Next, please. So we produced VP2, plant expressed VP2s by expressing the different VP2 of different BTV serotypes into a bacteria, Agrobacterium tumefaciens, and then doing its transient transform the bacteria with different VP2 and then expression in the tobacco plant, that is Nicotina benthamina. And you can produce here like the large amount of expression of VP2 gene, and you can easily purify them, and you can use them, these expressed VP2 protein, as a diagnostic candidate as well as a vaccine candidate. Diagnostic candidate we have already used, but in the meantime the project finished. We were waiting for the second round of funding, but we did not get second round of funding. So we have this VP2 expressed in this tobacco plant for use as a potential vaccine candidate for blue tongue viruses. Next, please. So this is the summary slide. I'm not going reading it. These are 6 different novel second, third generation vaccine platform that we have used. We have talked earlier throughout my talk, the different DNA vaccine, recombinant protein, chimeric viral vaccine, vector vaccine, RNA vaccine, and they are paving the way for advancement in human medicine. So they are there in the veterinary medicine, and these platforms can be or they are already being used and can be used in the human medicine. Next, please. So with this, I will like to finish my talk with the words of Edward Jenner, because we talked about vaccines. Never underestimate the power of a curious mind, because curiosity is the fuel that derives the innovation and progress. So thank you very much for your kind attention and hearing. And I once again apologize for the long time and the time waste. I sincerely apologize for this. I will look into the system. Thank you very much. And I'm happy to take any questions you may have. Thank you very much.
Thank you so much, ma'am, for your inspiring and enlightening words and sharing your valuable knowledge. I believe the participants will gain a lot of knowledge, especially in terms of immunology and veterinary sciences. We wish you all the best and sending you positive vibes and good energy for your coming days. Thank you, ma'am. And
Thank you very much, everyone, and Ms. Silo. Thank you.